So we're continuing chapter 7 on genetics and all that jazz. Life in the Petri dish. No, Elizabeth, no, don't go. Asexual reproduction. Cells making cells with the same genes in them, right? Offspring have genetic copies of the original. Now, in our bodies, those two cells might go on to do very different things. I mean, your blood cells are produced this way, but eventually it can produce red blood cells and white blood cells and platelets and all the different things that blood cells do. You have skin cells that produce hair. They use different genes. They may all have the same genes, but they use different genes. We're going to talk about the cell cycle. When a cell divides, a eukaryotic cell divides, it goes through a bunch of stages that are represented in this pie chart. And this green part is the part that's actually the division. Technically, it's the production of a new nucleus, which is generally followed by the two nuclei separating out into two separate cells. But just kind of a disclaimer, mitosis, which is the process we're going to talk about it like it's cell division, but technically it's nuclear division. And this pie chart, the way that it's divided up, isn't always going to be representing the timeline, where a cell spends all this much time between divisions and a really quick thing through divisions where all the different stages take the same amount of time. It's hugely variant in one cell to another. Now, a lot of the cells in your body are in this stage, and they're never going into that stage. Your nerve cells, once they're in place, they never divide again. Your muscle cells pretty much never divide again. Some of your connective tissue cells pretty much never divide again. So people go, oh, is there anything of the original me left after the cells have all been replaced? Yeah, there is. There's a lot of the cells that were there at the very beginning are still there. So this is mitosis. Yikes. So I'm going to talk about some of the stages before I write stuff down. This is basically the same representation but with a little bit of labeling. This is the division stage. This is between the division stage. Inter is a Latin word for between. Interstates were originally a between the states setup of roads. And a lot of stuff happens during this stage, that one of which isn't mentioned here because this is all about cell division and that's all they were concerned about. Those nerve cells that never divide again, those muscle cells that never divide again, those cells are doing their jobs. That happens during interphase, the phase between divisions, even if there's never going to be another division. So even in cells that do turn over fairly rapidly, like the cells along the lining of your digestive system, which turn over about once every day and a half, is that for most of that day and a half, they're doing their lining of the intestines job. So two major things happening during interphase for a lot of cells. Number one, their job. Number two, they set up a division if they're going to divide. That happens during the stage in between. The reason for that is once they start to divide, they've pulled all of their DNA tightly together so it doesn't tangle with each other. You know, all those different chromosomes don't catch on one another. And once you've got them all packed together like that, you can't really get at them to make proteins that you need. You need to have those all done and everything set up before you actually get into your division, is that you can move them around and activate the proteins that you made, but you better have them made because you're not getting at the genes to make them once the division actually starts. So, mitosis. Now, interphase isn't actually part of mitosis, it's between them. But I got to talk about it, so I got to put it up here on the list. So the regular job of the cell happens during interphase. And it prepares for division if it's going to divide. If it's going to divide, that's a big deal. When you have cancer cells, the division has gotten out of control. Cancer pretty much only happens in cells that divide anyway, that are regularly set up for dividing, because cells that are never going to divide again, they've got that stuff all shut down. It's almost impossible to activate it and set off cancer. That's why you get cancer in things like your skin and the lining of your digestive system and your bone marrow. If it's brain cancer, it's not the nervous cells in your brain that don't divide, there's support cells that divide in your brain. Reproductive tissues making cells all the time. So there's cancer that happens there. But you don't, when was the last time you heard about somebody having muscle cancer or heart cancer? Those cells don't divide, so they don't become cancerous. So what we have here, you can tag certain proteins with a compound that you can get to fluoresce if you hit it with the right kind of light. And so you can get things to show up in a cell that otherwise wouldn't show up. It's a type of staining, but it's a fancy type of staining. And that's what we have in these pictures, where they mostly have been staining it to make spindle fibers show up and DNA, eventually chromosomes here. This is interphase, and a few things are happening that I'm going to kind of gloss over. A lot of stuff happens. I don't expect you to remember all of it, but you're going to need to move the chromosomes around. You got copies. You got to get a set of copies to go one way and a set of copies to go the other way, so you can divide the cell between them and have a whole set of copies on both sides. To move those around, you need microtubules. Remember microtubules? Microtubules. Useful for moving things. Those microtubules, when cells divide, are called spindle fibers. It's just a particular type of microtubule. There's two little clusters, two little organelles that are going to, you get one from the last time the cell divided. Each cell gets one. They make a copy of that, and then they move the copies to opposite ends of the cell. And that designates the poles of the cell, like poles on a globe, which makes the middle the equator. So once they are over there, they start to grow microtubules out in every direction. This is the very first stage of the actual division, and it's called prophase. And a lot of stuff happens in prophase. It basically sets everything up. When I was learning the orders, you can remember IPMAT, but I always remember this, and I think it really should be prep phase, because that's kind of what it is. That isn't what it's called, but that's what it is. So in prophase, a whole bunch of stuff happens. Now, as usual, I'm not going to write absolutely everything down, so you're going to have to stay up with me. So you've got these two centrioles. They get a couple of other names too, but we're going to call them centrioles. And so these things are sending microtubules out in every direction. So I'm going to blind myself so that I can get into the middle of the screen. This screen is our cell. So here's a bunch of centriole microtubules going out in every direction. Here's a bunch. The microtubules on this side go over to the membrane and stick to it. Go over to the membrane and stick to it. And that holds these two centrioles in place. It anchors them. The other ones go out across the middle of the cell. Something else is going on. The envelope that surrounds the nucleus, these spindle fibers need to get in there and grab the chromosomes. You need the envelope out of the way. The nucleolus is integrated with chromosomes in a particular way. You need to get that out of the way. So the spindle fibers can get at the chromosomes. Then you need the chromosomes all packed up really tightly. So if you're watching this through a microscope, the nucleus, which usually has kind of an even stain because there's a whole bunch of DNA, but it's all, spun out so that you can use it. As prophase progresses, the nucleus starts to get stripy. Thin stripes and then thicker stripes, and eventually you start to see that those stripes actually are structures with two ends to them, and actually are two-ended things connected to each other. Sometimes. You can't always see that under a microscope, but that's technically what's happening. The chromosomes are getting more and more and more tightly packed together so that they are separate and they can't easily catch on one another. Sorry, worried about sound from behind me. So, I'm back in the cell. I am now one of the many chromosomes. I'm way too big, but so here's two chromosomes connected to one another at my, there we go, at my wrists. So the spindle fibers going back and forth and back and forth. So a spindle fiber comes over and doink, attaches to the centromere from this side. Once it attaches, it starts to shorten, it starts to reel it in. But a spindle fiber attaches from this side and starts to pull. Here's the rule. The longer the spindle fiber, the stronger the pull. So if it catches me over here, that one's longer than this one, so it starts to move me this way. Now this one's getting longer, this one's getting shorter. This one's getting weaker, this one's getting stronger. It's got to put me to the point where the pull on both sides is equal. It hauls the chromosomes into the middle of the cell. The equator. You need to get the copies away from each other. So first you got to get the copies so that you can move them around. Then you got to attach something to them that will move them. And then the easiest way to get them completely separate from one another is to line them all up before you separate them. And you don't really line them up, it's a three-dimensional line, but it always looks like a line from the side. And so this is all that happens during the preparation stage. So a whole bunch of stuff happens that sets up the next stage. Here's the nuclear envelope. Here's the nuclear envelope going away. In pieces, in bits and molecules, people disagree on just exactly where it goes. It probably goes different ways, places in different cells. Here's the chromosomes that originally show up when they're just stripes. They call them chromatin. Remember I said it's a confusing term? Because there's the original chromatin that you can see through a light microscope, and then the eventual chromatin that's just DNA structure in a nucleus that they discovered with electron microscopes. But when it first shows up and you can't see two ends to it, you call it chromatin. When you do the lab, you'll deal with that. Here's the Remember I said centrosome, centriole means the same thing, this is a centriole. Here's your spindle fibers. It would be nice if they put those on opposite sides like they have them here. I guess they're showing them moving into place. A lot of times they've already moved into place before this happens. So here's the nucleus, the envelope going away. Here's the nucleolus, it's gone by this point. Here's spindle attached to a chromosome, spindle attached to a chromosome. Here's a spindle heading for a chromosome. Here's one attached from two sides. And so they're trying to show a whole bunch of stuff in one drawing. And when we do it in the lab, some labs actually make you look for early prophase and late prophase. That's how much stuff changes over it. But we'll take anything as a picture of prophase for the mitosis lab. So the next stage is metaphase. And it's always represented as a line, but keep in mind, if this was a round cell, you'd be looking at the side. You'd have chromosomes all held this way in 3 dimensions. But once you get them all lined up, from the side they look like a line. So it isn't strictly a line, it's just, they're in the middle. Well, once you got them lined up, you're prepared to pull them apart. You just need them to go apart. And that's what happens in the next stage. I'll write this down. In an anaphase, the chromosomes pop apart. There's a chemical release that makes them separate. They used to think that it was a tug of war that pulled them apart, but it's not. And each of the 2 copies is attached to one of the 2 centrioles. So one set of copies goes one way, one set of copies goes the other way. Oops. Sorry. I didn't realize I hadn't written it down. Now don't make the mistake of thinking, well, metaphase, once they're in the middle, all they need to do is pop apart. That must go really fast. And you know, a lot of times it does go really fast, but it all depends on the cell and what the cell needs to do. And different cells, say you need to get ready, but you're not really ready to divide yet. Cells can stall themselves at almost any of these stages, including metaphase. They can hang out in metaphase for a long time. They can go through it really, really fast. Is that just because it looks like not much happens, doesn't mean that a cell can't stall in that particular phase. So the chromosomes are lined up. Then anaphase. The 2 copies separate. Now, classically, the stage after this is always shown as the cell dividing during it. But the thing is, the cell will divide, once it gets the chromosomes away from one another, it can divide as soon as it wants to or as late as it wants to. So this stage, cells divide in, the next stage, cells divide in. A lot of times they're all the way back to interphase and the nucleus is all back together, and then they divide the cells. Sometimes after a very long time, is that they can hang there with 2 nuclei in the cells for a long time before they divide. So there isn't, once you get into this, there isn't any magical phase where the cell always divides. We'll talk about the term for that, but it always is shown in this next stage I haven't written yet, but it doesn't necessarily happen just then. And here's an example. So in a plant cell, the chromosomes get away from one another, you get a space in between, the cell starts to build a new wall there. And that partial wall is called a cell plate. Animal cells, some of them pinch off now and form 2 cells. They don't have cell walls, remember, so they build the separation differently. You got the chromosomes away from one another. You need to rebuild the stuff that you had broken down so that you could get this to happen. You need to break down stuff that you had to build in order to get this to happen. So the stuff that happened during prophase kind of unhappens during telophase. Not necessarily a complete reversal of order, as long as it all gets done. The spindles need to let go of the chromosomes. The nuclear envelope needs to reform around the chromosomes. The chromosomes need to spin back out so they can be usable again. The nucleolus needs to come back. The spindles all are kind of pulled back in and broken back down to their bits and pieces. And maybe the cell divides now, and maybe it doesn't divide now. But if it divides, there's a term for that. So I'm putting maybe here. Cytokinesis. That's the term for a cell dividing. If you have a fish tank, there's a disease that you have to put like treatment in for. It's called ick. That's short for the genus name of the parasite Ichthyotherius. And the parasite is a single-celled animal, little protist. And it gets up and tucks itself underneath the scales of the fish, and it starts to go through mitosis. It goes through telophase, gets back to interphase, doesn't divide the cell, just does it again, and again, and again, and again, and again. So it goes to 2 nuclei, and 4 nuclei, and 8 nuclei, and it keeps going until there's hundreds of nuclei, and the cell has gotten big enough, you can actually see it peeking out from under the scales of the fish. And then, each nucleus gets its own cell. Suddenly cytokinesis happens, but it produces hundreds of cells all at once. And those cells drift down to the bottom, they take in nutrients from the bottom, and they grow, and then they go up and they reinvade the fish. So if you got a fish tank with not a lot of fish in it, you go from, oh, well, it's got a couple of spots on it, to, oh crap, the fish are all dying, because the reproduction in ick is exponential. But it's just an idea of cytokinesis works a bunch of different ways. In insect embryos, the first cell goes through mitosis until there's 1,000 nuclei all around the outside of the yolk in kind of a thin membrane layer. But it's all technically one cell. There's no membranes in between the nuclei until they hit a point. Membranes form between all the nuclei. It goes from being a 1 multi-celled embryo to a 1,000-celled embryo in one step. So the world works in a lot of weird ways. And then, of course, you're back around to interphase. And that's a good place to stop. We'll pick up meiosis in the next one.